I would like to introduce Dr. Oliver Hankinson, the Director of the Molecular Toxicology Doctoral Program.
Thanks, Miriam. I'm going to be very brief. I'm very pleased, as all of us in molecular toxicology are, to welcome back Julie, who was an alumna from our program, and Michael Roth, who was her mentor. Michael is a professor in the Department of Medicine at UCLA and a molecular toxicology faculty member. He was Julie's mentor, and he is going to introduce her.
Absolutely. Thank you, Oliver. I am very happy to see Julie come back, one of our own. She spent a few years here in our molecular toxicology from like 2010 to 2016. And she did great work while she was here with me, working on the biology of cannabinoid receptors and identifying and describing new subsets of cannabinoid receptors in immune cells and their intracellular trafficking and their role in B cells. And that work has been widely cited and I think really set a new insight into cannabinoid receptor signaling in biology. And then Julie worked for a few years in the drug discovery group at Amgen before moving on to the FDA, which I think she had her sights on for just since she graduated from our program here. You know, Julie was very actively involved when she was here in the Society of Toxicology, American College of Toxicology, and the Society of Neuroimmune Pharmacology. And I think, you know, that all that exposure and presenting abstracts and posters and her work, you know, led to the connections that eventually directed her interest into working in the FDA. And so certainly, you know, the type of exposure that we offer our students here can really make an impact on their career. And I think it certainly did for Julie, in addition to winning all the awards that she did at the society presentations, which is always nice, but, you know, leading to a career is really a great outcome from presenting work at scientific meetings. So I want to welcome Julie for her presentation. She's going to talk to us about toxicology and FDA-approved therapeutics. And also, I'm sure, tell us a little bit about her position that she currently has at the FDA. Julie, welcome. It's all yours.
Thank you, Dr. Hankinson and Dr. Roth, for introducing me and for inviting me back to UCLA. It feels great to be talking to UCLA students again. So I'm going to start the talk with a little disclaimer. So the opinions expressed by me do not reflect the official support or endorsement by the US FDA. So this is an outline of my talk. I'm going to walk you guys through the non-clinical, the research and development of a drug all the way from the bench to the market. Because I know when I was a grad student at UCLA, I didn't really know a lot about what it took to approve a drug, to bring it all the way from the bench to your house. And then I'm going to go through a couple of case studies as to drugs that I have seen that I've personally reviewed and how we dealt with the difficulties. So just a little bit of an intro. As Dr. Roth mentioned, I was a grad student of his. I completed my PhD in molecular toxicology from UCLA in 2016. I did research on the inflammatory effects of inhaled drugs of abuse. After graduating at UCLA, I became a researcher at Amgen in Thousand Oaks, California, and I did early discovery, COPD, and asthma research. And following Amgen, I took a position at FDA. I'm currently a toxicologist at the US FDA, and I work for the Division of Pharmacology, Toxicology for Immunology and Inflammation. So basically, we review drugs for various clinical indications. So I review drugs for pulmonology, allergy, critical care, rheumatology, transplant medicine, and most recently, COVID-19. So what is it that I do at FDA? So my role is I'm a pharmacology toxicology reviewer, and my role is to assess the safety of a drug based on the animal data and based on in vitro data, so based on Petri dish data. And so what we do is that we predict how the exposure and the toxicity that's observed in animal studies, how will that relate to humans? So we evaluate the non-clinical data. When I say non-clinical, I mean animal data. So we evaluate the animal data and how that's going to be used to support human clinical trials. And then we provide guidance to industry, so to pharmaceutical companies throughout the entire drug development process. And our primary goal at FDA is always safety. So we put humans first. So we are always looking at the safety of your drug or of your product. And so the main question that I ask myself when I'm doing my job is the dose that the pharma company is going to administer, is the duration of the drug, and is the route of administration supported by animal data? So I'm going to give you guys a little bit of a background on drug development 101. And just to emphasize, this is how a drug gets approved in the US by FDA. The vaccine approval is a very similar process, but it's also different at the same time, because with a vaccine, you're administered 1 or 2 doses versus a drug. You might take the drug every single day for the rest of your life, depending on your condition. So it's a fairly similar process, but in its own way has some differences. So you start out at the bench in a pharmaceutical company doing research, and anywhere from the bench to the market, it could take pretty much close to 10 years to develop a drug and bring it to the market. And it could be a process that costs over $1 billion. So you start out with research and development, you're at the bench. Basically, this is before a drug has entered a human. There's no humans involved. You're just doing Petri dish experiments and animal experiments. And so you start out by, first you got to identify a target. What do you want your drug to bind to? What do you want to study? Then you're going to develop an assay. You're going to do a high throughput screen because you need to have several candidates that might be able to bind to your target. You're going to get a hit. You're going to identify a hit. You're going to optimize, and then ultimately select the best drug candidate that you have. And then you're going to bridge the animal data to humans. And so what the pharmaceutical company has to do is they have to test their drug in animals, and they have to test it in vitro to determine, is this drug safe enough to be tested in humans? And also you want to know, does this drug work? Because you're not going to invest millions and millions of dollars if your drug doesn't work. So if you're claiming that your drug helps arthritis, you need to study that drug in an animal arthritis model and show me the efficacy. So once you have your drug candidate and you want to come to FDA because you're ready to start human clinical trials, what you do is that you file an IND. An IND is an investigational new drug application. So before you can file that, you can come to us for advice. So we offer something called a pre-IND meeting. So you can come to FDA and ask us questions. We're here to help. And so you can ask us, let's say, for example, your drug has a certain toxicity in animals. You see that it's increasing liver enzymes or something like that. You could come to us for questions and we can guide you as to how you can navigate this toxicity. What is the future of your program? And it basically gives us the opportunity to give the pharmaceutical company feedback before investing millions and millions of dollars. Okay, so you're ready to open your IND. You're ready to start your drug development process, basically. So most of the time when you open an IND, you start with phase 1. I'm sure with the whole vaccine approval, you guys have heard of the multiple phases of development. You've heard of the clinical trials are in phase 1, phase 2, phase 3. So generally you open in phase 1, which is a first in human study, but sometimes you can open it later in development. You can open it in phase 2 or phase 3. And that's mostly when you have clinical experience outside of the US. Sometimes pharmaceutical companies go to other countries to start their clinical trials because in the US, we are very, very strict and we require a lot for approval. Or also sometimes you might have human data because you're studying the drug in a different disease. So like, let's say, for example, you come to me and you want to study your drug in asthma, but you had already studied it in COPD. You can use that research to start your application in asthma. So once you submit all of your studies to me, I have to review everything in 30 days. I have a 30-day review window, and I ask for a lot of studies before you enter humans. So some of the studies that I review is that I review the pharmacology of your drug. So I review the primary and secondary pharmacodynamics. So basically, what does your drug do in vitro? And then also, is there any efficacy in an animal model? I also look at safety pharmacology studies. So I'm looking at how does your drug affect your respiratory parameters, mostly in animals? How does it affect your central nervous system? How does it affect your cardiovascular system? I look at the pharmacokinetics of your drug. I look at ADME. What is the absorption? What is the distribution? What is the metabolism? And what is the excretion of your drug? Also, does your drug form some sort of metabolite? So when you administer the drug, that's called the parent drug, and it might metabolize into your body into different metabolites. I need to know what those metabolites are, and you need to study those in animals. So I also review single dose toxicology studies. So you need to administer your drug to an animal. And this is mostly, you're going to do it at very high doses because the goal is to determine what is the maximum tolerated dose that an animal can take. So for example, what's the highest you can go without killing the animal or inducing severe toxicity? And also you need to determine what's the maximal feasible dose, because sometimes when you go really high concentrations, you might have some problems with viscosity or absorption. So you need to determine what's the highest you can go, what's the maximum feasible dose. And then also you're gonna provide me repeat dose toxicology studies. So like, let's say, for example, you want to start a clinical trial where you're administering a drug for 28 days. You need to test that drug in the animal for 28 days. And you need to give that to me before you can go into humans. And we require two different studies. We require a rodent and a non-rodent. And the non-rodent is generally a monkey, a dog, a guinea pig, a mini pig. So you also need to give me genotoxicity studies. So you need to tell me, is your drug mutagenic or does it induce chromosomal damage? And that's mostly in vitro studies. There's also other studies like local tolerance. So like, let's say, for example, you are injecting your drug. I need to know what is the effect around the injection site. The phototoxicity of your drug, will it make me more susceptible to standing in the sun? We look at absolutely everything. We also look at excipients. So when you are administering a drug, there's also several different ingredients in there in your tablet. It's not just one. So we look at every single ingredient that's in your tablet, because sometimes you might add an ingredient that makes it glide better, that makes it lubricate better. We look at absolutely everything. And then in your phase 1, most likely at this time, we don't know how drug affects fertility. So we look at that. So we are protecting the human population by asking men and female of women of childbearing potential to use contraception, because at this point, we most likely do not know how your drug affects fertility. So after the pharma company provides me with this laundry list of studies, what am I looking for in these studies? First of all, I'm looking at the study design. Did the pharma company conduct the study correctly? Did they use the right amount of animals? We have basically guidance documents, which tells the pharma company instructions as to what they need to do to conduct a proper experiment. So did they follow the guidance? Also, did they use a relevant species? Because sometimes monkey might not be the best species. It might not be the most relevant. Maybe you need to go into a guinea pig or into a lamb. So you need to provide justification that you picked the right animal. Also, most importantly, what is the toxicity profile of your drug? What are the side effects? And when we look at side effects, we look at, are they clinically monitorable or are they dose limiting? So when I say clinically monitorable is like, let's say, for example, your drug is inducing an increase in liver enzymes. We can monitor that in the clinic. We can just draw blood and measure your liver enzymes frequently. So we can monitor that in the clinic. So we say that that is not dose limiting, but we might have some sort of brain effect. We might have an effect on your central nervous system. That is something that we cannot monitor in the clinic, and it might be irreversible damage. So we say that is dose limiting. So that toxicity will stump the dose that you can go as high as you can go. So that is what we call dose limiting. So we look at the incidence. If we see a finding, in how many animals is this occurring? Is it just one animal or is it in 10 out of 10? We look at the severity. Is it just a very minimal finding or is it marked or is it severe? Also, what are the historical controls? Because it could be a background finding. There are times that there is something that happens in the animals because a lot of times they're under stress. So is this finding due to stress? And a lot of times the laboratories that are conducting these studies have a list of the background findings. So you're going to give me your historical controls to see, is this drug effect due to, or is this effect due to your drug, or is it just stress that the animal is going through? And then also, are these findings reversible? So like, let's say, for example, I see an increase in liver enzymes. If I stop administering the drug for 4 weeks, do those liver enzymes go back to normal? You need to tell me what is the reversibility of these effects. So the goal of the study is to identify a NOAEL. A NOAEL stands for a no observed adverse effect level. Basically, what is the highest dose that you can administer in the animal without producing a significant increase in adverse effects as compared to the control animals? So in this 30-day window, I have reviewed all of the data. I still have an opportunity to ask the pharmaceutical company questions. So like, let's say, for example, I see a toxicity that I'm not really familiar with. I have an opportunity to ask them questions and for them to correct the program. It's not just an automatic no. There is a back and forth that happens. And then once I have all my data, we do a safety meeting. So we meet with other disciplines. So it's not just me making the decision. It's not just the toxicologist. I'll go into a little bit more detail as to who the team is, but I'm consulting with physicians, with chemists, with statisticians. So it's basically 5 of us that have to say yes. It can't just be me. If I say no, the clinical program stops. If the statisticians say no, clinical program stops. So we all have to be on the same page. And so if we think your protocol in your program is safe, we issue a safe to proceed letter. You can officially start human clinical trials. So human clinical trials, if we're starting in phase 1 is generally healthy volunteers. They're most likely strong college students that have volunteered for this clinical trial, because it is a little interesting that you are volunteering for a drug that has never been tested in humans before. We're not going to start testing it in an elderly population. So these studies, there is mostly the intent is to study what is the metabolism of your drug, what are the side effects when you increase the dose, and then also we need PK, pharmacokinetic, and PD data, pharmacodynamic data, in order for you to move into phase 2. So once you're in phase 1, before you move into phase 2, I need you to give me more studies. So generally phase 2, you're going to do a longer term study. So like, let's say, for example, you started phase 1 with a 28-day study. And for phase 2, you want to move into a 3-month clinical trial. You need to test that drug for 3 months in animals. You cannot proceed to humans until you test it exactly the same in animals. And you need to do it in 2 animals. You need to do it in the rodent and you need to do it in the non-rodent. And then also you need to complete your genotoxicity studies. So the studies where you determined if your drug causes chromosomal damage or mutagenicity, you did that in vitro, you did that in petri. I need you to do that in animals now. So I need those studies before you move on to phase 2, and you're going to submit all of that data to FDA before you proceed to phase 2. So, okay, so you are ready for phase 2. What is phase 2? So phase 2 are generally larger studies. And these are generally in the patient population you're ready to study. So phase 1 was healthy volunteers, but now phase 2, you have a diabetes medication. Now you're going to study it in diabetes patients. And so these are usually controlled studies. They're dose range finding, because you are going to find what is the best dose for your drug, because phase 3 studies are very expensive. So you will not want to move into phase 3 with multiple doses. You will only want to move into phase 3 with 1 dose, the dose that works the best. And then the goal of it's also to determine what are the side effects of your drugs, what are the risks, and then for you to select the most appropriate dose for phase 3. So from phase 2 to phase 3, I'm going to ask for more animal studies. At this point, I need to know the effect of your drug on fertility. We don't know right now what it affects fertility, but before you move into phase 3, I need to know. So you're going to do animal studies. So you're going to do animal studies that assess the reproductive and the developmental tox. So you need to show me what is the effect of your drug on fertility and early embryonic development, and then how does the drug affect embryo fetal development. Then also, if your drug has metabolites, you need to provide the metabolite studies. And then also juvenile animal studies. So like, let's say, for example, you want to study your drug in a pediatric population, you need to give me baby animal studies. You cannot do them in adult animals. You need to do it a juvenile animal study, because at the end of the day, children are not just small adults. They're very different. They're in a development stage. So you need to prove to me that the drug is safe in a juvenile animal. And then so before, as I mentioned before, phase 3 is a very expensive stage. So you can come to us and you can request an end of phase 2 meeting. And this is basically where you get to meet with us and we tell you, okay, your animal studies are complete. We have determined the toxicity profile of your drug. And then also, these are the studies that are going to be used to bring your drug to the market. So you're going to submit all of this information to FDA before you move into phase 3. So phase 3 is what we call for marketing authorization. These are the clinical studies, the human trials that you are going to use to bring your drug to the market. And so what are these studies? These studies are generally long-term studies. They're generally anywhere between 6 months to 2 years that someone is administered a drug every single day for 2 years. And so this is where we determine, does your drug work? Because we don't only assess safety. We also need to know, is your drug efficacious? Does it work? We want to know overall what's the benefit risk profile of your drug, because we use all of this for labeling. So when you get a drug from CVS or Walgreens and you open your little cardboard box and you see that there's a big insert in there that's like multiple pages long, and most people don't even read it. If you stop and read the insert, that's where all of the data is of every single study. So if you want to know what is the effect of your drug on fertility, like, can I take the drug when I'm pregnant? Just read the insert, because all the animal studies and the animal data is in there. So the studies that I require from phase 3 before I bring it to the market is, I need to know, does your drug cause cancer? You need to do carcinogenicity studies, and you need to do it in 2 species. You need to do it here. It's most likely a rat and a mouse. And these studies are very expensive studies. So you need to be serious that your drug works and you're prepared to bring it to the market. And then also, I need one more reproductive study. I need to know, how does your drug affect postnatal development? So like, let's say, for example, you're pregnant and you take the drug. How will that affect your fetus, and how will it affect the development of your baby? I need to know that. So you're going to do that in an animal study. And so then before you proceed to attempting to bring this drug to the market, we call this, you file an NDA, a new drug application. You can come and meet with us before you do this, because once you file an NDA, there's a fee. So the fee, I checked this morning, what's the latest fee? The fee is $2.9 million. So you have to pay $2.9 million for us to review all your data and determine, can you bring it to the market? So that's a very expensive fee. So we allow you the opportunity to come to us and meet with us for a pre-NDA meeting, so we can talk about everything. We can talk about, is your drug ready? Because it will be a huge waste if I tell you, hey, you're missing one monkey study, you didn't do that. So we give you the opportunity to get all your ducks in a row. And then once you submit the new drug application to me, I have 1 year to review the data. I have 12 months. The clock starts where I have to review thousands and thousands and thousands of pages of data. And this is where we work on the label. So we work on that little insert that's in your cardboard box when you get a drug. We work on that. We work on that with the pharmaceutical companies. We're going back and forth with the drafts, because it's an agreement between what they propose and what we propose. So after I've reviewed all these studies that I've mentioned, I have to make a decision. Do I approve this drug or not? And once again, it's not just me that's approving the drug, but I'm mostly going to be approving the drug based on the toxicity profile that I saw in the animals. Let's say, congratulations, you have a drug that is efficacious and safe, and you have brought your drug to the market. You're not done. There's phase 4. So phase 4 is what we call post-marketing. So just because you got your drug approved, doesn't mean that we have forgotten about it. We are studying this drug for a long time after its approval. because now the drug has been in the hands of a lot of people. So now we need to know, are there any side effects that we didn't see in our controlled studies? So phase 4 is mostly for the physicians. It's mostly analyzing the clinical data, but there are some toxicology experiments that I might need. So for example, if I approved your drug for adults 18 and older, but you want to start in a pediatric population, you got to give me those juvenile animal studies. You cannot move into a pediatric population until I have determined that the drug is safe in juvenile animals. So then you do all of these follow-up studies and you submit it to FDA. So now that you know a little bit about drug development 101, what it takes to bring it from the market or from the bench to the market, what is it that I do? So I review the studies. I'm reviewing these 1,000-page study reports, and then I'm writing. I'm always writing. I'm writing memos, I'm writing reports, I'm doing presentations, and then I also am receiving consults. So my area of expertise is lung. So any inhalation drug product comes to me, comes to my group. But let's say, for example, you have a new drug that's inhaled insulin. That's going to go to the group that specializes in diabetes, but they're not inhaled experts. So most likely they will consult with us because they don't know what's a normal lung effect or not. So we do a lot of consults. We also do a lot of meetings. We meet internally with our team, and then we meet externally. We meet with pharma. Some of the resources that I have available to me, because really no job prepares you for the job that we do at FDA. We are a regulatory agency, and honestly, no one else approves drugs other than FDA. If FDA says no to a drug, it's no. So we are a regulatory agency. So no job that you've done prepares you for the work that we do. So we have a lot of training. We mostly work one-on-one with team leaders, which is an unofficial boss that we have, and then we work with our supervisors. We also have subcommittees of experts. So for example, if I have a really, really interesting toxicity that I don't know how to interpret, there are experts for everything at FDA. For example, I had a drug that was affecting the habituation in rats. What is habituation? Habituation is your ability to get used to something. So like, let's say, for example, with your clothes, if you had something that wouldn't make you get used to how tight it was around your neck, how tight your jeans are around your waist, it would drive you crazy. You know, eventually you get used to something. And I had a drug that was affecting your ability to get used to something. It was really bizarre. And I have no idea about anything about that. So I had to consult with the central nervous system experts because I don't know, I'm a lung expert. So that's the great thing about FDA. They have experts for everything. We also have a lot of courses. We take a lot of courses. We're always learning. We're always staying up to date with the latest in science. We have a lot of workshops, and then we have guidance documents. Guidance documents is what I mentioned, that these are like the instructions that we have for the pharmaceutical companies, that we basically write it out to them. We write it out that this is all you need to do to approve a drug. So we're following those documents. Okay, I've mentioned several times that there's a team of people. It's not just me. So who is on my team? So my team consists of a regulatory project manager. That's the person that is reviewing the application. And also they mostly communicate with the pharmaceutical company. I don't do a lot of interacting with them on a daily basis other than my meetings with them. So if I need to write something, I send it to them and they send it to the pharmaceutical company. Then the physicians, we work very closely with the physicians. They're the ones who are looking into the human data. We work with clinical pharmacologists. They are the ones that are looking at the drug disposition in the body. They look at your drug concentration and the exposure, because generally, if you increase the dose of your drug, you should increase the exposure of the drug. They look at drug-drug interactions. So let's say, for example, you're on medication and you want to know, can I take an aspirin while I'm on this medication? That's the job of the clinical pharmacologist. And then they also look at how certain subpopulations react to the drug. So for example, if I have some sort of kidney impairment, I can't break down this drug as well as someone has a perfectly functioning kidney. So they look at the subpopulations that they might need a lower dose. And then the statisticians. the chemists. The chemists are the ones that look at everything that has to do with manufacturing and then the controls. So as I mentioned earlier, we look at every single ingredient in your drug. So if you're adding a glidant, a lubricant, we look at all of that. Then we also look at the container. So if you have a drug that's in a vial and the vial is sitting on a shelf for 2 years, we look at the amount of stuff that's leaching from your vial into your drug. We look at that. We look at the cardboard box where your vial is stored in. We look at absolutely everything. And then ultimately we work with the statisticians. The statisticians are the ones that are telling us, are these effects significant or not? Because we might see a side effect in 0.00025% of the population. And they're the ones that are in charge of telling us this is a significant side effect or not. Okay, so now that you guys know a little bit about drug development and a little bit of what I do, these are a couple of case studies of drugs that I had and how I dealt with these issues, which was very important because I've been at FDA for I think 3 or 4 years now, and so I was a very new reviewer. So they were really interesting issues that I faced in my first couple of years. So in case study number 1, I had a drug for a rheumatological condition. So like, let's say psoriasis, arthritis. And the pharma company was coming to me that they wanted to do 2 clinical studies. They wanted to do a single dose. So they wanted to administer the drug, 1 dose to the humans at a time. And then they wanted to do a multi-dose study. So they wanted to administer the drug every single day for 28 days to healthy volunteers. And this was a first in human study. So the drug had never touched a human before. We had absolutely no idea how the drug was going to behave in humans. So we relied entirely on the animal studies. So when I looked at the animal studies, I saw that the kidney, there were some side effects in the kidney. The kidney was identified as a target organ. And this was based on tissue findings that I saw in a 28-day study in rats and in monkeys. So the side effect was observed in both rats and monkeys. But I determined these side effects are not dose limiting, they're clinically monitorable because we can monitor for these kidney effects in the clinic. But there was something really weird about the drug that I didn't really know how to interpret. So the exposure of the drug didn't really follow the classic dose response relationship. So when you increase the dose of your drug, you're exposed to more, so your exposure should go up. And this drug wasn't really following that clear pattern. So we suspected that we were undercharacterizing the toxicity profile of the drug. So we are very strict on first in human studies. We look at everything in so much detail because we have no idea how the human is going to react. So we sent information requests to the pharmaceutical company saying that, hey, we know that there's this problem with your drug. It's an unknown pharmacokinetic activity because it's never been tested in humans. So we're going to ask you to not do that multi-dose study. You're only going to do a single dose. So what you're going to do is you're going to administer 1 dose to the human, and then you're going to come back and report to me. And that's the only will allow you to proceed. And so they agreed to do their single dose study, and we allowed them to proceed. So I thought everything was great. A couple months later, I get an email. 8 out of 8 people in your clinical trial are severely sick. What do? Immediately, we place the study on clinical hold. We do not allow them to proceed. We do not allow them to dose any more humans because we need to stop and investigate what happened. So the 8 out of 8 people that got sick were, they had kidney issues. And as you guys remember, I had identified some kidney issues in the animals, but what happened in the clinic was very different than what we saw in the animals. And so this was one of the rare cases where the animal studies were not predictive of what happened in the clinic. Because, at the end of the day, we're not the same as a dog. We're not the same as a monkey. The animal studies can only go so far. So this was one of the cases where humans were way more sensitive to this drug than the animals. The kidney toxicity that we saw happened at much lower doses than what we had seen in the animals. So we stop and pause here to think, how do we get this clinical study up and running again? How do we determine what's a safe starting dose? So we took an approach called a MABEL approach. MABEL is a minimum anticipated biologic effect level approach. So basically, everyone in the team got together, the physicians, the clinical pharmacologists, the toxicologists, we got together and we came up with this approach. So the pharmaceutical company need to come up with some sort of modeling, some computational modeling to predict what is the lowest dose that they can go that will give nearly imperceptible side effects. And so once you propose this super low dose, we're also going to need way more renal monitoring. It's going to be a very strict renal monitoring. And the pharmaceutical company agreed to do this. They agreed to lower the dose to almost imperceptible effects and the very, very, very strict renal monitoring. And they were allowed to proceed. So for case study number 2, I have a different drug. And this time it's for a pulmonary condition. So asthma, COPD, seasonal allergies. And the pharma company was coming to me that they wanted to do a single dose protocol. So administer the drug one dose at a time, and then a multi-dose protocol where they wanted to give the drug every day to a healthy volunteer for 14 days. This was once again a first in human study. The drug had never touched a human before. So we rely solely on the animal data. And so in the animal data, I saw, okay, there's a cardiac effect. I see that there are changes in hemodynamic and ECG parameters, but we determined that this is not dose limiting. It's clinically monitorable. We can monitor for this in the clinic. We saw an elevation of liver enzymes in the 28-day monkey study. Once again, we can monitor this in the clinic. Then I saw some tissue findings in bone and in the liver of the rats. The liver findings in the rats, monitorable, not dose limiting, as long as they're not severe. But the bone findings are what really made me stop and think. I saw bone necrosis findings and effects on the turnover of your bone. And so we thought, this is potentially dose limiting. I don't think we can monitor for this in the clinic. It's not like I can go ahead and take a bone necropsy, right? So I had to stop it, and I had to say, this is not my area of expertise. I need help. So I consulted with the bone experts. So I consulted with the Division of Bone, Reproductive, and Urologic Products. The pharmaceutical company was saying these bone findings are not relevant to adults because we're no longer growing. So it's not relevant to us. But the bone experts said otherwise. They said, honestly, bone turnover takes place throughout the entire lifetime of an adult. So it is relevant to adults. So we can't just go ahead and ignore this. But we are not as concerned because we saw the bone findings in a 28-day rat study. We didn't see bone findings in the monkey. And this is why we always look at 2 species to see, are we seeing it in both species? Here, we only saw it in the rat. We didn't see it in the monkey. And we didn't see it when the rat was administered a single dose or 14 days. We saw it when it was administered 28 days. So when it was a longer term study. So the bone experts concluded the concern overall is low for a 14-day clinical study. So we sent the pharmaceutical company a request that we said, we see these bone findings. We don't know what to think about them. It's a first in human study, so pretty strict. So you're just going to do a single dose study. You're just going to administer 1 dose at a time and come back to me. You are not allowed to proceed with the multi-dose study. And then you're also going to have to monitor for bone turnover biomarkers. You're going to have to exclude patients with any condition that might affect bone turnover. And you're going to have to monitor for those cardiovascular effects that I saw and the liver effects that I saw. And once they agreed to that, the study was allowed to proceed. Okay, so the last case study that I have is a COVID-19 case study. As you guys know, that we've been in a pandemic for the last couple of years, and most of the COVID-19 applications came and fell in our lap because we are the lung experts of FDA. So for this particular application, we had an inhaled drug product for COVID-19. So the pharmaceutical company came to us with a phase 3 clinical study. They were starting right off the bat, phase 3. And they were proposing a multi-dose clinical trial for 14 days in hospitalized COVID-19 patients. So why were they starting in phase 3? Because they already had human experience. They had already been studying this drug and for other respiratory conditions outside of the US. So they had a lot of data. They had a lot of safety data. So it was correct of them to start in phase 3. So I looked at the animal studies. This was a monkey study. And I saw that the lung had some side effects. The lung was identified as a target organ of toxicity. I saw some findings in the 14-day monkey study. But at the end of the day, we determined that these findings were not dose limiting. These findings might have just been the body reacting to an inhaled substance. We said that these findings were possibly due to anti-drug antibody formation. So like, let's say, for example, you inhale something and it makes you sneeze. You're just sneezing because your body's reacting to it. It doesn't necessarily mean that what made you sneeze is toxic. So for these side effects that I saw in the lung, it was just the body reacting to it. So we said these side effects are not dose limiting. So we allowed the pharmaceutical company to proceed with their clinical trial in COVID-19. So a couple months later, we get a notice from the pharmaceutical company that there are these massive worldwide shortages that they need to switch manufacturers. We know that there's shortages for everything right now. Every gift you can think of, there's shortages. We even have a shortage of monkeys. We don't have enough monkeys to do more studies. So this pharmaceutical company, they need to switch the manufacturer. They can't stick with the old manufacturer, but the manufacturing process is different. So potentially, this is a brand new drug. If you can't guarantee that the manufacturing process is the same, it's potentially a new drug, and you might be introducing new risk to these patients. So this is not my area of expertise, so I consulted with the chemists, because the chemists are the ones that are controlled. They're in charge of all the manufacturing, all the controls. So the question here is, would we support a change from manufacturer A to manufacturer B? And the chemist said, no, you need to do a comparability assessment. You need to assess whether manufacturer A and manufacturer B are indeed the same drug product. And then I say, if the comparability assessment says the drugs are not the same, then I need more animal studies. I need you to give me an animal study with manufacturer A, manufacturer B, and do they have a similar toxicity profile? So as of right now, the pharmaceutical company can't continue with their studies until they determine that these drug products are the same. They're not allowed to start in humans, and they are unfortunately at a halt right now. So that was the last of my case studies. So thank you so much for listening to me talk. And I will pause here for a Q&A.
Great, thank you so much. That was a really excellent talk. So I would like to, or Christina, do you want to pause the recording?